Hello and welcome to today's webinar. I'm Danielle Dumont, the Marketing Communications Manager with YSI, which is now part of the Xylem company. Thank you for joining us, and today we're going to examine how combining measurements of several types of microalgae in water, such as chlorophyll and cyanobacteria, will provide more accurate estimates of total algae biomass. Our presentation will take about 35 minutes and we'll cover these topics. You'll learn about why it's important to measure algae and how it can be done in the field. After the presentation, we'll dive into your questions for about another 10 minutes. So at any time throughout the live presentation, go ahead and submit your questions or your comment by typing it into the questions box that you see on the side of your screen. Now I'd like to introduce our presenters. So first we have Brandon Smith, and Brandon is the Assistant Product Manager for the EXO Sond line. He joined YSI last year. Also we have Tim Finnegan. Tim is the Product Manager for the EXO product line, and he's been with YSI since 2002 in a variety of engineering and sales roles. And with that, we'll begin our presentation, and I'll turn it over to Tim.
Great. Thanks, Danielle. I think a good place to start is why scientists, researchers, monitoring groups, and you folks online really may want to monitor algae biomass. I've come up with three reasons as a starting point. First is that photosynthetic organisms are really at the base of the food chain. So if we can understand how these microorganisms behave in natural environments, it's a very useful starting point for a monitoring program. Second reason is the fact that all photosynthetic organisms contain chlorophyll A. So measuring chlorophyll A fluorescence is a very good indication of the phytoplankton biomass, and that directly correlates to the health or ecological status of an environmental water body. The third reason is the fact that some phytoplankton can be harmful through producing toxins or taste and odor causing compounds in drinking water sources. Now when you have an algae bloom on top of that, we can exacerbate these issues and present other challenges such as triggering low oxygen conditions, physically damaging fish gills and other aquatic animals, and generating large masses of decaying biomass. So now that we all want to measure algae, let me start off with some of the basics. You've already heard me use some of these key terms, but I want to take a moment at the top to explain some of the meanings that we're going to use throughout the presentation. So if we start off with algae, it's a very broad term, but it commonly has two different classifications. We have macroalgae, and this is the larger aquatic vegetation and plant matter like seaweed, kelp, and pond scum. And then there's also microalgae, and as the name implies, this is the microscopic photosynthetic organisms in the water. Through photosynthesis, this microalgae is responsible for close to 50% of the atmospheric oxygen production. Now our sensors are focused on measuring this microalgae, and we use two different channels. Now, some of this microscopic algae lives on surfaces. Others float freely throughout the water column, and these mobile algae are known as phytoplankton. Our sensor measures phytoplankton biomass via chlorophyll A fluorescence. And now there's an enormous diversity in phytoplankton, and there's different classifications, sizes such as diatoms, dinoflagellates, chlorophytes, the list goes on. And they take on a variety of size and forms. One thing that's absolutely unique amongst all of this diversity is the fact that all photosynthetic, and they're all photosynthetic, and therefore they contain the primary photosynthetic pigment, chlorophyll A. Now our final two terms are cyanobacteria and blue-green algae, and they generally are used interchangeably. However, cyanobacteria is a more precise name. Cyanobacteria are photosynthetic form of bacteria, and they can also take on many shapes and forms. Please note we'll be using these two terms interchangeably throughout this webinar. So before we start talking about field measurement, I want to walk through the process for lab sampling chlorophyll A through extractive pigment analysis. There is a lot of confusion around users around differences between in situ chlorophyll fluorescence with the field sensor and the process I'm going to review here, which is the lab extraction of chlorophyll A from a field sample. And by the end of this presentation, we'll make that abundantly clear. So the standard method can be broken into 4 simple steps. The first is you go out and you take a water sample of sufficient volume. Then you concentrate that sample, typically through a filter medium. You extract the chlorophyll through the use of an organic solvent like acetone or ethanol. And then you take a reading, use a spectrophotometer, an HPLC, or even a lab fluorometer, and you use standard method to generate qualitative chlorophyll A concentration data. So some obvious limitations of this approach are the labor, the time, the cost of collecting and analyzing all these samples. It can also be really difficult to capture a bunch of high-resolution data, which means you could miss natural dynamics and short-term events that can occur really rapidly. But the big advantage of that measurement is you get very accurate quantitative readings. Now if we shift gears and look at in vivo measurement, there are some differences from what we just discussed. In situ sensors offer the ability to monitor algae biomass in real time and at relatively low natural concentrations. Now some manufacturers call this in vivo chlorophyll measurement. Some use the term chlorophyll A, and we simply use the generic term chlorophyll. But in principle, they're all the same. All of them are underwater fluorometers tuned to optimally detect algae through chlorophyll A fluorescence. Now, when you make in vivo measurement, it's really important to remember there's a large diversity in phytoplankton size, shape, health, and even how the chlorophyll is packaged. Despite the diversity, most of these phytoplankton will respond to the chlorophyll excitation signal. Now, if we look at a cyanobacteria cell on the left specifically, there's a very significant difference in how this chlorophyll A is packaged in the cell and how the photosynthetic system is designed. So the result is that although cyanos have chlorophyll A, they don't produce a large fluorescent signal when the chlorophyll A is excited by the sensor. So what we do is we use a second excitation channel that optimally excites cyanobacteria via something called their accessory pigments. And these are found in the thylakoid membrane shown in the upper left. By detecting accessory pigments in cyanos and the chlorophyll A in phytoplankton, we can accurately estimate the total algae biomass. So our solution is to use an in situ fluorometer to estimate algae biomass in real time through chlorophyll A fluorescence by exciting at 2 different channels, one targeting cyanobacteria, the other targeting all other phytoplankton. We need both because the difference between prokaryotic and eukaryotic cells, but we're going to stop at this level of detail. Brandon will discuss how this measurement is made in the technology section. Here I just wanted to outline why we need to measure both. So let's move next to try and interpret some data. On the screen here, I have data from a YSI 6 series sonde on Grand Lake St. Mary's that was collected last summer. The Ohio EPA and the Ohio DNR have a really good monitoring program nearby. Now the most obvious part of this data is the short duration spike in cyanobacteria levels as measured by the cyanobacteria probe. This indicates an algae event. And because of the short duration of this event, it is likely it would not have been captured under a periodic sampling program. Under the right conditions, algae can reproduce very quickly. Now in this case, the cyanobacteria signal, which is shown in blue, went from reading 25% to reading all the way up around 125% during the algae bloom. If we look at the purple line, we're looking at the supersaturated oxygen levels. They reach almost 250%. Now naturally, oxygen levels will fluctuate around 100% in natural water. However, in this bloom event, cyanobacteria are converting light into oxygen at a rate faster than the oxygen can diffuse from the water. You can also see a significant impact on turbidity, which is shown in green. As the cyanobacteria clump together, they will increase the scattering of light and thus cause an increase in turbidity. This is why the turbidity, the cyanobacteria, and the DO readings all spike together. Another interesting point we really wanted to make with this webinar is shown by looking at the algae data through the 2 excitation wavelengths. The chlorophyll channel in red and the cyanobacteria channel in blue vary greatly in the same water. red chlorophyll fluorescent remains relatively constant even during the large cyanobacteria bloom. In this case, if we had only measured algae with a single wavelength, we would have missed the algae bloom completely. This is why it's so important to really excite both through the chlorophyll and accessory pigment channels so that we can properly estimate the total algae biomass. And a quick point, you can see a small diurnal type cycle in the cyanobacteria sensor, and I'm going to let Brandon cover this photoinhibition effect in his section a little bit later. I'll pass it off to Danielle now so we could actually get some of your input and keep this somewhat interactive.
Hey, good. Thank you, Tim. So right now we've launched a poll that you can participate in. There are several ways to measure algae, and we'd like your feedback to know if you are currently measuring algae concentration or algae fluorescence, and if so, with which method. So go ahead and, if you would, please vote in the poll, and we'll leave it open for a few more seconds. Tim, we also had a question on that graph. Do you know the time scale that was the x-axis for that? Do you recall?
That's a very good point. That was over about 90 days worth of data. Take that back. That's over 60 days of data collection. Sorry about that.
Nope, that's okay. And the y-axis was measuring, it's in percent.
Well, it was scaled because I have so many parameters on there, so I just kind of normalized it. And some of them were reading percentages, some of them were reading levels. So the signal reading was really running from 0 up to about 300 was the top of the axis. And I'll have that data set continued through this webinar.
Okay, good. Okay, good. We have some good results on the poll, and I think everyone is just about finished logging in. So I'm going to close this poll and share those results real quickly. So as you can see, the grab samples in the lab analysis are right now the most common method for our audience to determine algae concentration in their water. So that'll be good. We'll have good information coming up about how you might do that with field data collection. So in the next few slides then, Brandon's going to go over a new field sensor that uses fluorescent measurements from both chlorophyll A and accessory pigments in the blue-green algae. And take it away, Brandon.
All right, thanks, Danielle. So YSI is a leader in developing sensors for continuous monitoring systems. We have algae sensors tuned to excite chlorophyll and accessory pigments in cyanobacteria. And our latest platform, EXO, combines these measurements, but our legacy 6 Series platform supported these measurements separately. Both of these platforms, EXO and 6 Series, measure algae in the same way, through fluorescence, as Tim has described earlier in the presentation. We're emitting a particular wavelength of light to excite a chlorophyll A molecule. This molecule then emits a light of lower energy, and in this case it's red, and our sensor collects that fluorescent light. In the sensor diagram on the left, you can see a drawing of the EXO Total Algae Sensor. Behind the probe's window, 3 optical fibers exist. 2 direct orange and blue excitation light into the water, and 1 collects the incoming fluorescent light and directs it to a photodiode, a light detector. The photodiode then converts this fluorescence into an electrical signal, and that in turn is our raw data. Depending on how you calibrate the instrument, the sensor can give 2 outputs. It can convert this data to a 0 to 100 scale and RFUs, or relative fluorescent units, with 0 as no chlorophyll and 100 as a relative maximum chlorophyll value. Or it can also convert the raw data into an estimate of chlorophyll A and phycocyanin pigment concentration in units of micrograms per liter. And we'll talk a little bit more about this when we dig deeper into calibration later on in the presentation. The Total Algae Sensor for EXO is only one of 2 algae sensor options that a customer can purchase from YSI. With 6 Series, YSI offers 2 separate probes for exciting chlorophyll A and the accessory pigment. But the underlying functionality is similar to what we've just discussed with EXO. Using these parameters still remains a valid option for existing 6 Series customers who might want to add these parameters to their data set. Comparing our legacy sensors to the EXO Total Algae Probe, though, EXO's Total Algae Probe has many advantages. The most apparent is that both cyanobacteria and chlorophyll A excitation channels are combined in just one probe, so it frees up an additional sensor port for another parameter. And this combined design also has the added benefit of generating a more accurate estimate of the total algae biomass in the water due to its ability to simultaneously detect fluorescence from both phytoplankton and cyanobacteria, as well as its improved in sensitivity to degraded pigments and other common interferences like turbidity or fluorescent dissolved organic matter. The total algae probe also can detect lower concentrations of chlorophyll with higher resolution and with faster response times compared to its predecessor's 6 series model, while its titanium housing also allows it to be used deeper underwater and makes it insensitive to corrosion. But while measuring chlorophyll and blue-green algae together is necessary to get accurate data, as we saw from our St. Mary's example, it's also important to understand how an algal bloom is affecting overall water quality. Measuring other parameters like dissolved oxygen, pH, and temperature concurrently can paint a more detailed picture of the health of an environment. An EXO 2 sonde equipped with the total algae probe still has room for 5 more probes and a central wiper. And the platform's wiping system is probably a necessity for taking in vivo measurements within waters with higher algae concentrations to keep the sensors clean. Compared to our legacy sonde's anti-fouling solutions, EXO's central wiper can extend a long-term deployment for up to 90 days in the field, and that's sampling every 15 minutes or so. If you power the sonde externally, it could last even longer depending on the extent of the environment's biofouling potential. And oftentimes I hear customers discuss the importance of high accuracy data, and at YSI this doesn't just apply to our sensors' performance in a lab. It means high performance in the field where you actually need it. So on the right, we have a test done by ACT, which is the Alliance of Coastal Technologies, and it compares our 6 series legacy equipment to competitors. And as you can see, while the other instrument performs well initially, as time passes, its inability to counteract the biofouling that occurs means the data becomes virtually useless. So now if you compare our sonde, the 6 series sonde, over the duration of the testing, you can see that we have high quality data over the entire period. So imagine if this test were then performed with EXO instead, with its improved anti-fouling technology. Now I'll go ahead and pass it back to Tim so he can discuss some of the major applications for measuring algae.
Thanks, Brandon. When we started, I spoke about the roles algae plays at the base of the aquatic food web. But there's other practical uses for this sensing technology, including early warning of algae blooms and the ability to trigger samplers while blooms may be occurring. It's also very popular in drinking water reservoir management because the sensors provide the ability to provide high-resolution data at a really low cost. So I'm going to start off by talking about eutrophication applications. Now, eutrophication is the aging of a water body, specifically the accumulation of organic matter in a water body over time. It's a natural process. However, the accelerated eutrophication is a widespread problem due to anthropogenic impacts. These impacts are due in a large part to an increase in nutrients, such as nitrate and phosphate, that are triggering increased algae growth. Now, there's 4 problematic reasons associated with this algae growth. The first is the fact, is one I touched on earlier, and that is some algae species produce toxins that make the water dangerous. So this can obviously impact human and animals ingesting the water. The second problem is the increased algae can lower oxygen levels. This happens once algae die and decompose. The microorganisms devour this dead algae, producing anoxic water conditions. The third issue is a consequence of the last, as these low oxygen levels can suffocate the fish. And the last problem is the fact that algae diminishes the light penetration into the water. This occurs because planktonic algae absorb light before it can reach the bottom, and then this in turn it reduces the productivity of submerged aquatic vegetation. Now, because these events are becoming more frequent, so is the need to monitor these through sensors to help quantify the magnitude and frequency of these problems. Our algae sensors are very commonly used in drinking water applications because the data they generate really helps improve treatment efficiency. So a typical use would be to profile with a multi-parameter sonde throughout the vertical water column. If algae is detected at a certain level, an alternative intake may be used to draw water back to the treatment plant. When monitoring the remote source water for algae, you can also take preventative steps to reduce the cost of treating water. For example, if water high in algae is coming in from the raw water reservoir, the activated carbon levels and filter media can be optimized for when the water arrives back at the treatment plant. And most importantly, with a baseline of data over several seasons, understanding of when the reservoirs will turn over, when blooms may occur, and how long to run mixers can all be extrapolated from the water quality data. So it can be a real effective tool for reducing algae and taste and odor compounds caused by algae. In most of the major drinking reservoirs in the United States, the vertical profiler system is currently being deployed on sondes, measuring algae, turbidity, and DO throughout the water column 24/7. With that, I'll pass it back to Danielle so we can actually learn a little bit about what applications you guys are currently monitoring algae in.
Great, thanks Tim. So here's our second poll that we would like folks to participate in. So feel free to give us your feedback out of the many environments where algal growth may be of a concern to you. In what application are you currently measuring, or are you interested in measuring algae biomass? And we'll leave this poll open for a few more seconds as folks weigh in. And Tim, one of the questions that came in while we're waiting on the poll, people want to know if the EXO sonde with the algae sensor can be used only as a continuous monitoring device, or can it also be used to take some sampling measurements?
That will be a great segue into how we calibrate it. And really there's two ways to calibrate it, and that impacts if you're going to use it for continuous or spot monitoring. So we'll talk about that as the webinar continues. Great.
Good. Okay, looks like most of the folks are finished voting in the poll, so I will close the poll. And the predominant application is natural waters, as we can see in rivers, lakes, and streams. So lots of good feedback there. So as we move along in the webinar, we're going to have a few more slides that will discuss algae sensor interferences, and then as Tim mentioned, we'll get into some calibration of the algae sensor. And first we'll hear from Brandon.
All right, thanks Danielle. So as Danielle mentioned, in this last section of the webinar, we'll touch on some of the interferences with in situ algae measurement, and we'll also dig into calibrating EXO's Total Algae Sensor. As I discussed earlier in the webinar, the Total Algae Sensor is insensitive to interferences due to aspects of its optical design, but it is important to note that these interferences, while minimal, do still exist. There are three common interferences that affect the quality of your algae data: turbidity, FDOM, or dissolved organic matter, and temperature. As you can imagine, turbid water and water with high quantities of dissolved organic matter can absorb and scatter light emitted from our sensor and prevent it from reaching the algae cells. Additionally, the problem is twofold because these factors can inhibit the fluorescence from reaching the sensor's photodiode, the light detector that I mentioned earlier. Here on the right, you can see the improvements we've made with our optics and electronics to reduce these effects dramatically. As the FDOM content, or the fluorescent dissolved organic matter content, and the water increases, the corresponding fluorescence measurements are minimally affected. You can visually appreciate the advancements from our legacy system by noting the scale of the two graphs. We pride ourselves in the fact that our legacy sensors were some of the best in countering interferences. And as you can see, our team has reduced the FDOM interference by an order of magnitude. On the left, we can see the inverse relationship with water temperature and sensor readings as well. EXO automatically compensates for changes in temperature through our temperature conductivity probe. But if you're using other products, this might not be the case. Finally, another fluctuation you might see in your data concerns chlorophyll pigment degradation. Speaking at a very high level, in situ chlorophyll fluorescence will fluctuate depending on the light levels in surface water. This variation is unrelated to algae biomass, but rather is just a natural result of decreased chlorophyll fluorescence under high ambient light levels. This effect is called non-photochemical quenching, and you can see this effect in the data from any in situ chlorophyll fluorometer deployed in surface waters, with a slight decrease in readings during the day. Ultimately, the answer to how to combat interferences that I've discussed is to be informed on how other parameters impact your data, and then to measure them as well. It's nearly impossible to infer ecosystem health by measuring just a single parameter. So as I mentioned, calibration is a topic we get many questions about, so I'll go ahead and pass it back to Tim for an overview.
All right, I want to go through two different approaches to calibrating these sensors, and I think this will apply partially to the question about using it versus continuous and spot sampling. Now, as you remember, our sensors are making an in vivo measurement in the water. As a result, the only primary standard available to calibrate these sensors is a water sample itself. So in this way, the sensor is different from other sensors on a multi-parameter sonde that may have primary standards available, an example being something like a pH buffer. So this is not the case with algae sensors. And even though reagents exist for calibrating lab fluorometers and spectrophotometers, these are all extracted pure chlorophyll A reagents where the cell walls are not intact. Where with in situ, we're taking a measurement that excites the whole cell. So there's two approaches to calibration. The first is driven by groups that are interested in getting qualitative trends. So the advantage of this is its low cost and its high speed of calibration. It involves frequent one-point calibrations with filtered DI water and occasional second-point calibration with a secondary standard to adjust for any probe drift. The second approach outlined on the right requires collecting field samples and the use of extractive analysis I discussed at the beginning of this PowerPoint. Then these lab results are used over time to develop a correlation with the sensor output. Now, once a correlation is developed for a specific site, it can then be post-processed or calibrated more accurately to reflect the algae or chlorophyll concentration. This will help you get absolute values for microgram per liter readings instead of just RFU percentages. So the challenge with using this in spot sampling is you may have to develop different qualitative associations for different sites. So we'll keep that in mind, and I'm going to talk first about the qualitative or the relative change approach. Now, the first out-of-cal box should be a 2-point cal as you take the sensor for the first time. What this does is it sets the slope of the sensor. And when you're doing the 2-point cal, the first cal point is a filtered DI water, and that's representing zero fluorescence. And in the software, we select the RFU unit Brandon described, which is a percentage output from 0 to 100. And we accept the cal once the sensor shows that it's stable. Now, for the second cal point, we have a recommended rhodamine dilution as described in section 511 of the manual. And what this does is it serves as a secondary standard. Basically, a 0.625 milligram per liter concentration of rhodamine dye is approximately equivalent to 15.8 RFU, or 15.8%. And again, to calibrate, once the software shows stable, you accept this calibration and complete the process. And now we're going to go and log data in RFU percentages at all of your sites. And the process is very similar for cyanobacteria sensor. And throughout the course of use, you're going to want to routinely zero the fluorometer and check for drift at the second point, just to ensure good overall qualitative data across all of your sensors and all of your sites. And because the calibration resides inside the sensor and EXO, if it's moved to a different sonde, it will retain that calibration. Now, I'll go into 2 points here. If we were using this in a sampling application at many different sites, this qualitative approach is beneficial because it's going to give you a percentage RFU reading, and that's going to be standard across all the sites you use it at. So if you're sampling, the first starting point is to get some of this qualitative data. And that would be able to show you that this site is maybe at 20%, this site is at 50%, and that's a very big difference. Now, the data set, here I did put a scale, and that was a question that came up earlier. So we're showing RFU percentages. And on this data set I discussed earlier, the chlorophyll and cyanobacteria lines are shown on separate plots. And you can clearly see the cyanobacteria signal changing by 50%. And chlorophyll levels are remaining relatively stable. Now this gives us very good trending information. In continuous monitoring, we see that trend over time. If you were sampling, we could compare those percentages at different sites. Now I want to move to the more advanced calibration process. And we still need to start with some baseline data. And I'm going to use the same data set we looked at before. Now the next step is going out and collecting field samples for extracted lab analysis. And so ideally you want to go out and sample at different concentrations, under different flow conditions, at different seasons in the year, at each site that you're doing your monitoring at. And so we've represented this by drawing some circles over the data set. And of course this is a lot easier said than it is done. But it's really critical to take these samples at the same point the sonde is measuring using a sampler. So now let's assume we've collected a number of samples and we want to develop a relationship between the sonde reading and the lab reading. So what I've done is put some of this data in Excel for my different samples. And in column B I have all of the RFU values from the sensor. In column C I have all the water samples I collected and that were analyzed at the lab. And what I do is I develop a relationship. I used the data analysis tool pack, and Excel generated a second-order polynomial. And this converts all of my RFU readings to microgram per liter readings. Now, most of our users actually stop here, and they just post-process all of their RFU data to microgram per liter using this regression line. And the fit of this regression line is largely driven by how dynamic a site is and how many samples you collect. And going back to the question that was asked, if you were using this system to sample at different sites, it is very likely you would have to develop a regression or fit or take these samples for each independent site. Because I have to stress that each site can be different and it can change with seasons. And as you collect more samples, you're going to develop a stronger correlation curve. Quantification can also be difficult because samples are not always very homogeneous. Now the last step that a small portion of our users go is they then take that data and instead of post-processing it, we can put some of that correlation data in the sonde and calibrate it that way. So what we do is instead of using the RFU channel, like we talked about earlier, we're going to choose the microgram per liter channel. And this will help complement some of that post-process data and allow a real-time output of semi-qualitative value. So I take the regression line from the previous slide, and I go ahead and prepare my secondary standard just like I did before, a 0.625 milligram per liter concentration of rhodamine dye. The difference is, instead of using our default microgram per liter value from the manual, I'm actually going to calculate the microgram per liter value using that regression line. So what I've done is I've taken my rhodamine dilution, which is 15.8%, I've pushed that through my regression, and it says that that's equivalent to about 24.29 microgram per liter. I can then calibrate my sensor that way. So instead of using the RFU, I calibrate under microgram per liter, and I use 24.29 microgram per liter. And that's going to give me good qualitative data for that site based on the samples I took. Now I know that was difficult to describe through this webinar, but that does describe two different approaches to using the algae sensor to get qualitative versus quantitative data. Now algae is extremely important, and it's becoming more important with increased algae events. And EXO makes monitoring algae really easy. There's a lot of technology built into all the sensors and the software that automates a lot of what we talked about. And with the anti-fouling technology, you can get really good high-resolution data over a longer period of times. And if you're using this in a sampling application, it's great because you can measure over 12 independent parameters on a single platform as you go from site to site. So it is a really powerful tool for understanding the environment. Now I want to apologize. We have a lot of people on the line and a lot of questions coming in. I'm going to do my best to try and run through some of these, but I'm going to ask for or apologize in advance. It could take us up to a week to kind of sort through all of these and get back to you. And some of them we might confer with our microbiologist and get some help for some of the advanced users on the line. So please go ahead and type those questions in, and apologies in advance because I know we are not going to even be able to get to a lot of these just because of time. So Danielle, I'll pass it over to you.
Good. Okay. Thank you, Tim, and thank you, Brandon. And thanks, everyone, for your participation so far. As Tim mentioned, there's some good questions that are coming in. And so we'll take about the next 10 minutes and go through some of those questions. And then, as Tim mentioned, if we can't answer your question live today, we will get back to you next week, probably through an email or phone call, to be able to address what you're interested in knowing. Just a few more notes. Also, if you are a current EXO customer, you currently have an EXO Total Algae Sensor, there is factory training at the YSI office in Ohio on April 23rd. That comes as part of the purchase of your SON. So if you're interested in attending that and get some hands-on training, there's more details on our website about that. Also on the website, you can register for future webinars, and you can also access recorded webinars like the one we did last month on FDOM. So for the next few minutes, Tim or Brandon, are you guys ready to answer some questions?
Sure. I have a couple questions I can answer real quick.
Okay.
So thanks, Danielle, by the way. So we got a couple questions here about probe maintenance. So this one just asked, do the total algae probes require much maintenance? So no, there's really very little maintenance involved with the probe because of its optical design. So you just need to keep the optics clean to get good readings. So just before you take it into the field, inspect the lenses occasionally, and if there's anything that you need to clean off, use a really non-abrasive cloth, probably something lint-free if cleaning is necessary. Another question we have here is someone that's asking more of a definition type question. So they ask, what is the difference between phycocyanin and phycoerythrin, which are some terms that Tim discussed in the presentation. So both of these, at a very high level, they're both accessory pigments for cyanobacteria. So phycocyanin is the major accessory pigment in most freshwater cyanobacteria species, whereas phycoerythrin is the major accessory pigment for the marine species. So EXO's Total Algae Sensor comes in two versions, one for customers who might work mainly in freshwater, and another that work primarily in marine or estuary environments. So the only difference between the two is that the excitation light for the accessory pigments is tuned a little bit differently.
Great. Just some simple questions here. First of all, one was a question how people can access this after the webinar, and you'll get a link, and this is available online. So if you want to review this presentation, it's at ysi.com/webinars. You can get a recorded copy of this. Another question that came in was about the graph. And generally, every time I show that same graph with chlorophyll response and cyanobacteria response, all of that data was recorded in percent readings. So when you saw a low line that was reading around 5% on the chlorophyll, that was relatively stable. Now we are always exciting in taking a chlorophyll A measurement, although in the data you saw, there wasn't much response of chlorophyll even during the cyanobacteria bloom. And so that's why the chlorophyll line was relatively flat, because the majority of the algae biomass was coming from cyanobacteria. So I hope that clears up a couple questions I'm seeing in here. Now, one was about how are interferences and adjustments made. A lot of that's really through new optical filter designs in the sensor itself. So the optics and electronics have been improved in EXO pretty substantially. And also because these are all digital sensors now, the optics are really almost attached directly in a lot of cases to the A to D processing board. So we're able to reduce a lot of the noise levels as well. We have another question.
I'll go ahead and jump
Yeah,
in
go
real
ahead.
quick, Tim, while you look at that. There's so many questions here. So I'll tackle this one. So this is from a 6 series user. Can the total algae probe output data in cells per milliliter like my 6 series probes? So this is a question relating to our legacy equipment. So the legacy equipment did in fact have a data output and units that were cells per milliliter. But no, the new technology doesn't. And when we developed that, we did that intentionally. And this was because at the cellular level, when you're looking at our fluorescence sensor, there was so much noise. So we decided to not include that in the new version. But if you are interested in still having these units, you can convert them if you establish a relationship between the sensor's fluorescence and the algal cells per milliliter unit in a post-process step.
Great. One question came in here about calibration, and this is one thing that is actually pretty exciting. They talked about, do you have to swap the sensor out every time you calibrate the zero? And what's nice about EXO is you can actually fill a cup with 0 DI water and then go through and calibrate all of your zeros at one time without removing those different sensors and channels. In fact, even if you had 2 algae or 4 algae sensors, you could put them all in one EXO sonde, and you could calibrate 0 on all 4 of those probes, and each of those probes has 2 channels. So you could go and calibrate 0 on 8 channels without having to refill that cup in this example. Now, another question, there's several in here about FDOM or CDOM impact on readings. And we just had an FDOM webinar that talked about color dissolved organic matter and our sensor, which is a fluorescent dissolved organic matter. And the challenge with DOM is that it can change throughout the seasons. You know, as leaves and tannins get in the water, that can change the color of the water. And that can affect, you know, how several different sensors can operate. So what a lot of people are using an FDOM sensor for is to measure the organic matter levels and see how they're changing over time. So the question comes up about interferences. Well, we've designed the probe to optimize it or minimize the effect of DOM on the readings. But as you get very high concentrations, or more importantly, as your concentrations of DOM change over time, you kind of need that FDOM signal so you can understand if that is biasing the reading at all. Because it's very likely there's times where you have very low organic matter levels, and there'll be other seasons where you might have very high levels. And that would tell you if it's having an impact on any of your other sensor data. So it's a little bit difficult to say precisely, but that's generally how it works and why we use a separate sensor to see if it is having, if the concentrations are changing highly.
All right, I've got a couple questions here, Tim. I'm seeing a lot of questions asking about pricing for both the probe and the sonde. So pricing is really highly dependent on your sonde sensor combination for whatever application you might need, and it'll be very specific to your needs. So it's better to request pricing through our website, and that's exowater.com. You'll see it on the next slide if you need to see it. And on the home page, there's a request pricing button on the left side. So go ahead and click that, and we'll get back to you immediately with any pricing concerns.
I have a question here about how long is the life of the probe and what is the maintenance? Now, it is an optical probe. It does have a 2-year warranty that covers it. And because it's all laser welded shut, it is completely sealed and submersible down to 250 meters. Of course, the expectancy of that probe is substantially longer. Easily, you could expect to, in typical applications, use that sensor well beyond that 2-year warranty period, perhaps 5 to 7 years without issue. And the maintenance side of it is incredibly low maintenance. Because it's an optical probe, it doesn't really drift. So all you really have to do is keep it clean, and that's about the extent of the maintenance. People use a second point rhodamine dye check just to verify that the sensor hasn't drift or the LED hasn't started to burn out.
So I've got a couple questions here asking about more specific sensor specifications. One in particular is asking about the detection limits for chlorophyll. The detection limit for chlorophyll is 0.09 micrograms per liter chlorophyll. And if you have any questions or after this you need to check sensor specifications, just again visit the website. That's exowater.com/sensors. You can also look at the accessories, anything that you might be interested on that website as far as product information is concerned.
A question here came in about, does the exosonde automatically adjust for temperature changes of the rhodamine dye or stream temperature? So the point here is, when you're doing the calibration, you need to use the lookup table and put in the appropriate temperature adjusted value. But then while the sensor is deployed out sampling or taking measurements, the onboard temperature sensor, the CT probe, the conductivity temperature probe, will automatically temperature adjust the output reading while you're taking the reading. And this is the same, for example, with pH. When you calibrate it, you need to put in the correct temperature value from the bottle or from the lookup table. But once it's calibrated and deployed, the temperature will automatically adjust. A couple questions about data logging and wireless. So if you're just doing continuous monitoring, there's a few ways. You drop some batteries in the sonde and you set it up typically for a 15-minute logging interval. It'll wake up, take readings, and store it in its internal memory. And now memory is inexpensive. There's lots of memory on board. And then I'd say about half of our users actually take sondes and hook them up to data loggers so you can transmit that data wirelessly over the web. And again, lots of questions about calibration. One was saying, hey, if I go and take samples at one of my reservoirs and do all the lab analysis, will that apply to my 3 or 4 other reservoirs? That's difficult to answer without some of this data, but what you do is you need to take a couple samples to validate that. So you develop a correlation curve for one reservoir, and then go and take a couple spot samples at the other reservoirs and see how closely they come to your predicted values. If the dynamics, the concentrations, the speciation at each of those sites is consistent, which it very well can be, then it's likely your calibration correlation curve will be consistent across those reservoirs. But there's also cases in, say, a river system where several miles apart within the river system, you have very different species and different correlation coefficients that apply. A question came in about the windows. All of the windows are sapphire windows, so they're really smooth. They clean very well, and they're very difficult to scratch or change. Yeah, sorry, there's hundreds of questions in here from all of you. Thank you so much for sending these in, but we're filtering through some of them. Yeah, so here's one that came in saying, hey, I'm currently just looking at, I'm only using a chlorophyll sensor now. And it sounds like you're better off measuring both. And that's absolutely the point we want to make is, if you're only looking at chlorophyll A sensors, you can miss a very big portion of the total algae biomass. And so the plot I kept showing was a perfect example of that. The chlorophyll sensor was flatlined, and we were having huge algae blooms. And that's because those algae blooms were primarily cyanobacteria, and we picked those up. And the vice versa can be true as well. You can have algae blooms from phytoplankton that aren't cyanobacteria, and that would be detected on the chlorophyll sensor. So if you're interested in monitoring algae, we really feel it's very important to measure both of those channels so that you get a very good estimate of the total algae biomass.
Tim, Brandon, that was about 15 minutes of questions.
Okay. Well, yeah, I would say in closing, we have a lot we'll go through here, and please give us some time, and we'll just have one of us or a microbiologist respond to you via email personally for those that we didn't get to. I apologize.
So if you have any last-minute questions that maybe you haven't thought to put in, go ahead and add them to the list, and again, we'll get back to you.
Good. Well, thank you, Brandon. Thank you, Tim. And thanks, everyone, for your participation today. This webinar has been recorded, and so it will be available for playback within the next few days. If you've Since you have registered, you will automatically receive a follow-up email from us, and that will contain the link to the recorded webinar. And as we mentioned, we'll also get started on answering the questions that you've submitted today, and if we didn't answer them live, then we'll be able to follow up with you